Imperial Yeast is at it again with their Imperialis project, creating yet another unique proprietary strain through hybridization. In addition to its excellent attenuation and rapid reduction of diacetyl, I-10 Mangostini contributes robust ripe tropical fruit, strawberry, and lychee notes that complement modern hops. And as a Kveik hybrid, it can be fermented anywhere from 78 to 90 degrees Fahrenheit or 26 to 32 degrees Celsius without any negative issues. Head over to imperialyeast.com to learn more about I-10 Mangostini and be sure to pick some up for your next batch of fruity IPA. Welcome to the Brew Lab. Free amino nitrogen sounds kind of like a health supplement that we'd see on a TV commercial during a sporting event. Take your free amino nitrogen supplement, boost your energy, and focus. But it's actually not that far off, to be honest. Free amino nitrogen, or FAN, is kind of like a health supplement for yeast. I'm your host, Cade Job, and today in the lab I'm speaking with Dr. Glen Fox, the Anheuser-Busch Endowed Professor of Malting and Brewing Sciences at the University of California at Davis. Now, 2 weeks ago on the episode with John Palmer about beer maturation, we discussed how setting the conditions for a vigorous and thorough fermentation was one of the keys to helping your beer mature quickly and fully. Things like having enough yeast nutrient like zinc and free amino nitrogen can not only improve attenuation, but maturation as well. So this conversation with John and others that Jordan and I have had on the show made me realize I need to better understand FAN. What is it? Where does it come from? Why do we need it? Is there a point where enough is enough? Can you have too much? And of course, the perennial brewing question, how do we control it? I'll ask Glen all of those questions, and we'll also get into some tips and tricks about how to increase or decrease FAN. And we'll spend a good chunk of time on things like FAN that survives to packaging and why you may want to start paying more attention to the FAN that makes it into your beer. And once again, a big thank you to our patrons. If you're not currently a patron, I encourage you to head to patreon.com/brewlosophy and check out the monthly contribution levels. In addition to the feel-good vibes of helping to support the work we do here at Brewlosophy, you get access to a bunch of awesome rewards depending on the different pledge levels that you choose. You get access to contributor recipes that we've never before published, new discounts each month to YakimaValleyHops.com, and for $3, access to a monthly live Q&A session with a special guest from the brewing industry. We've had guests like John Palmer, Jamil Zainasheff, Vinny Chilurzo, and even Mr. Brewlosopher himself, Marshall Schott. And when you sign up, all of those prior sessions become available to you on our private Patreon Facebook page. So please head over to patreon.com/brewlosophy to find all the information you need to get access to this awesome resource. Thank you to everyone who's left a rating or review of the show. If you haven't yet left a rating or review, I'd love for you to do so. We're so close to 100, and I'd love to get some more reviews to get us over that mark. Also, reviews are a great source of feedback for me. It helps others like you find the show, and without those reviews, we, of course, wouldn't have the Applying the Science series with Jordan, and the show wouldn't be what it is today. So thank you to those of you that have left a review, and if you haven't, please take a moment to help us out. Feedback is brought to you by the clever folks from North America's leading hop supplier, Haas, who developed Incognito, a 100% all-natural hop product crafted to deliver highly concentrated flavor while maximizing brewing efficiency and reducing process loss. You wanted more varieties, and the Haas team heard you. Incognito is now available in 8 of the most popular Whirlpool hop varieties, such as Centennial, Chinook, Citra, Equinaut, Eldorado, HBZ 472, Mosaic, and Sabro. All the hoppy goodness you expect without adding any actual hop matter means less process loss, increased profits, and best of all, more beer. You can learn more about Incognito and all the other innovative products Haas has to offer at johnihaas.com. That's John, the letter I, Haas.com. All right, listener Alex wrote in about episode 130, using gene editing for hop breeding. He said, this most recent Brew Lab episode came out at a perfect time for me. My dad's visiting and requested a brew podcast in the car to see what they're like. He's a research leader for a maize, soybean, and barley breeding program at the USDA, so his eyes lit up listening to Cade draw a comparison between corn breeding and hop breeding. He doesn't drink much, but he's got 100 questions about the USDA's hop program. So thank you, Cade and Jordan, for convincing my dad that beer and Brewlosophy is hip and neat. Awesome. Thank you, Alex, for that wholesome feedback. And I'm glad you got to share a moment with your dad over that episode. That's one of my favorite parts of this podcast, touching on research from other areas that might influence the way we think about beer or brewing, much like your dad with corn, soybeans, and barley research. There's an amazing amount of overlap. Thank you again for sending in your feedback. And if your dad wants more info, just send me his contact info and I'll forward it along to Chris Willig. All right, I'm going to take a quick break and I'll be back in a few minutes with Dr. Glen Fox talking about free amino nitrogen. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Firm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brewlosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app. On several different episodes, there's been mention of this thing called FAN, or free amino nitrogen. It's something that most brewers have at least heard of, or at least are aware of, but being honest, it's something I know very little about, and it's a question that's come up on quite a few episodes, so I figured we need to get somebody in the lab to talk about it. So thankfully today, in the lab with me, I've got an expert on FAN, Dr. Glen Fox of the University of California at Davis. Glen, welcome back to the Brew Lab.
Thanks, Cade. Great to be back and chatting to you about a really interesting topic.
Yeah. Well, and listeners will, of course, recognize you from episode 2 of the podcast. It's hard to think that was almost 2 years ago, and certainly 130 episodes or so, 134 episodes since we were last on there. We talked about MASH methods and IR spectroscopy, but I think people will also recognize you as the principal advisor for a couple of other guests on the show. James Bruner, who came on to talk about his research with hop creep and yeast, and Sophie Held, of course, coming on talking about beta-glucans and enzymes during the MASH. And there's been a lot of great research coming out of your lab, and I've enjoyed having you and your work looking at a range of brewing topics, including proteins, which I think are a little bit relevant to what we'll be talking about today.
Thanks, Cade. And really, when we start talking about FAN, some of the information that we'll discuss actually is related to mashing methods. So it's all of these things tie together.
Yeah. I love that about brewing, don't you, right? How everything just seems so interrelated. I mean, we talked about that with James and Sophie. I mean, their research especially, two totally different things, but still dealing with different aspects of enzyme activity.
I always enjoy when the students do the senior brewing class and they think, oh, well, this should be just a lot of fun. But we challenge them to think about the deep chemistry and biochemistry that's going on. And we had a midterm last week, and a number of the students came up afterward and said, this is a lot more complicated than we really thought. So it's sort of
right.
The underpinning of all brewing is how complicated it is, and it just makes it even more challenging but rewarding.
Oh, gosh, yeah. And it seems like it gets more complicated, but also as things crystallize, right? As you start to understand what is FAN, you can start to understand what to do about it, and we'll get into some of those things today. But an introduction of you as well. So you're the Anheuser-Busch Endowed Professor of Malting and Brewing Sciences at the University of California at Davis. Of course, got your PhD from Southern Cross University in Australia. Published over 100 peer-reviewed articles, and recently co-authored the revised Scientific Principles of Malting and Brewing Science, which is a textbook that you're likely using in your brewing class, and I know that is used around the country. I've asked you before how you came to study brewing science, but I'll ask a similar yet different one on this episode. How did you come to the position at UC Davis, and what have you guys been up to over there?
Well, I probably came into the industry through a little side door, and it was probably having more of a background in grains and malting science that may have appealed to the faculty who ended up making the decision that I get this position. And it was really a long-term understanding of the UC Davis program, I'd actually sent some of my students from Australia over to UC Davis to work with Charlie on different topics. So I had a long-term sort of relationship with Charlie through the brewing industry. And because I knew how well this program was structured and the value it was having to industry, I saw an opportunity to set up something similar in my university in Australia. The craft brewing scene is still growing in Australia, probably similar or if not slightly better percentages than the current craft scene in the US. But there's also this significant gap in education and understanding, and all the craft breweries are always saying, we're looking for people that have got a good understanding of the process, maybe not high-level experience, but at least a good understanding of what's going on. And then when this job came up, actually Charlie was in Australia way back in 2017, and I got him to come and talk to my colleagues at the University of Queensland to talk about the pros and cons of setting up such a program. And everyone left thinking, well, this is an opportunity. And we sort of followed on from there, and I got some internal funding to get a decent-sized pilot brewery, and that was ordered just before I left in July 2019. And then something happened shortly after that, so they couldn't deliver the brewery until the end of 2020, early 2021, and it sort of sat in the pallet. And then 2022, they managed to set it up, but I was only back there in September a couple of months ago to do some early brewing trials on that system. So it was nice to see that come to fruition after a couple of years of planning, and they're already planning training courses for undergrads as well as industry.
Oh, that's fantastic. That's fantastic. Like leaving a mark there on those. And yeah, I certainly understand, right? I mean, a lot of things shut down in 2020 and 2021 especially. But it's true, right? I mean, your comment there about helping helping brewers learn and understand the process. It's just so important. I mean, and we mentioned earlier, there's so much that's going on and so many things that are happening. I mean, goodness, there were 136 episodes into a podcast that focuses entirely on brewing science. So all kinds of things to learn and dive into. And of course, what we're going to talk about today then is free amino nitrogen. What is free amino nitrogen? Where does it come from? What does it do? Why is it important? Why do you need to care about it? How do you increase it? How do you decrease it? How do you control it? I don't know that we'll be able to cover everything in an hour, but we'll certainly give it a good college try. Sound good?
That sounds good. We'll have a good crack.
All right. Sounds good. All right. Well, we have to start then at the very front, right? Which is, what is free amino nitrogen?
So easily we just say FAN, but free amino nitrogen is the proper terminology. And this is basically the amino acids that are free in malt and in wort, usually produced partially in the malting process, but that may actually be added to the total amount of FAN during the mashing process, depending on the mashing conditions. But it also includes dipeptides, or two amino acids still bonded together, or maybe even a tripeptide, which might consider three amino acids bonded together. It just depends on the method and how we measure FAN, but we're really talking about the base units of proteins. The amino acids make proteins, which include the enzymes. So these are absolutely critical in the malting process. We're possibly still going through some hydrolysis of proteins by proteases in the mash. And then we're producing this sort of profile of these free amino acids that will be required by yeast during fermentation. So that's sort of generally what we would consider FAN to be. It's basically these hydrolyzed proteins coming from the grains and then producing more amino acids in mashing.
Yeah, so it's sort of like, I like the way you described it. It's kind of like the building blocks for a whole bunch of things, right? It's the building blocks for proteins. It's the building blocks for enzymes, right? We've talked a bunch about enzymes. I mean, of course, like I mentioned earlier, we talked about with James and both, right? James and Sophie both are looking at enzymes, and a building block of those things is amino acids. And so free amino nitrogen are essentially free building blocks. And I like the important point there too, right? Is it's not just an individual amino acid building block. It could include dipeptides or tripeptides as well. So not just that amino acid. And you were touching briefly there then on like the malting process and how that involves and impacts FAN. Where does FAN come from?
The malting process is just hydrolysis of these large substrates. So again, trying to not dive too deep into the biochemistry, but we would consider starch, which is made of glucose, as a major substrate that will provide the fermentable sugars for yeast. And the storage proteins in barley and wheat and other grains that are used are basically the substrates that will provide amino acids in these peptides and dipeptides and tripeptides. So again, this is an enzyme system that's breaking down these substrates. Now, the malting process is a very live process where we're synthesizing these enzymes. So we need these amino acids as the building blocks to make or have these new proteins synthesized. During the mashing process, there's no more synthesis of these enzymes. This is where we're really testing the survival rate of these enzymes. And some mashing systems are just too hot for these enzymes that would continue to hydrolyze or break down the proteins. So we lose potentially more FAN based on mashing temperatures. The really high mashing temperatures around 150 Fahrenheit basically limit any more hydrolysis and freeing of amino acids. The next live portion of our process is in fermentation. So basically the end of mashing, lautering or separation, then boiling, there's no more enzyme systems working until we get into fermentation, which again, the yeast will be synthesizing enzymes as part of a number of different pathways to produce alcohol and produce other things that the yeast does. So there's these two, or basically the malting and the yeast are the two major systems where we're synthesizing enzymes. And the mashing process is really just extracting what has been hydrolyzed. And the boiling process basically is making sure all those live enzymes are denatured. But just mentioning the boiling process, you asked about where these things come from. It's probably something that a lot of the brewing industry doesn't consider when they're adding hops in the kettle, and certainly when they're dry hopping, and James would have touched on this, is the hops have proteins and amino acids as well. So when we're adding hops, we're potentially adding additional sources of amino acids. The profile of amino acids is different to what we would see coming from the grains. And then we're dry hopping, we're adding new sources of amino acids to present to the yeast, which the yeast may or may not care about at that time, depending when you dry hop. So it's probably one of those things that we don't give enough credit to the hops in terms of things it's contributing other than the alpha acids and maybe some aroma compounds. But the hops are contributing more as well.
Yeah, that's interesting. So, it really sounds like FAN is just, coming in throughout the process, right? It's starting in the malt house. It's changing during the mash, boiling. We didn't really mention that, but again, that's, concentrating of wort and, that sort of stuff. But then fermentation and then hops and dry hops. So it's kind of coming in throughout the process. And I remember back to this, you mentioned earlier about like starch, and Sophie did a great job of describing what beta glucanase does, right? Like that enzyme. She described it as kind of like opening a package, like for Christmas or something. You've got wrapping paper around a cardboard box, and then inside is the present, and the present is the starch. That's the sugar. That's what we as brewers want. But you've got to get to it first. It's not, the starch isn't just floating out there, ready to be hydrolyzed.
Certainly not. No, this is really a complicated biochemical series of steps. So the analogy, and Sophie's used a great analogy, but another one to think about is actually a building with thousands of rooms, and every room is a distinct cell. And within that cell are starch granules, and surrounding those starch granules is the storage protein. So we have to, Sophie talking about breaking down those walls, the glucanase will attack the granule, which is the beta-glucan, which is the structure of those walls. And then the proteases could come in and start to hydrolyze the protein, the storage protein, which acts as a sort of barrier for the starch granules being attacked by the starch degrading enzyme. So it's almost like this inhibitory effect where these hordenes and glutellins, which are the storage proteins, acting as somewhat as a barrier. It's the last stand, if you can imagine,
Yeah.
of resistance for the starch to be hydrolyzed. But it's these hordenes and glutellins that are the main proteins that are going to be hydrolyzed that will contribute to FAN. So depending on the level of modification of malt, then these FAN levels can change significantly, because under-modified malt might actually not have as much of the cells exposed for the proteases to attack the proteins. So the level of modification is important, and then the mashing condition to measure the FAN also can influence the final level of free amino nitrogen.
Yeah. I mean, it's just such a good way to describe that, right? That it's coming through, and that those hordenes, those storage proteins, that's what's being broken down. And during the malting process, we're sort of priming the enzyme package, right? So we're not only getting access to the starch granules, but we're setting things up for the brewers to have success in the mashing. And so everything's getting changed and like you mentioned, broken down. And so those hordenes are actually breaking down into those fragments of FAN, free amino nitrogen. Now, I think most people are aware, certainly we talked about it with Harmony Bettenhausen a few weeks back, talking about malt COAs, and FAN is a metric that's on the malt COA. And I find it interesting here, because we're talking about, well, malting seems to play a big role, but it sounds like mashing also has an influence. It may be coming in during fermentation, either through an exogenous source or through the yeast themselves, and then hops might have it. I guess I don't know that there's a question here. It's maybe more of a statement. Is like, it surprises me that we just are looking at FAN from a malt perspective as a brewer, and then sort of maybe just hoping that FAN does, or that we have enough, or that it makes things it does the things that we need it to do once we've got it into the brew house. Am I off base there, or is that kind of how brewers may be approaching FAN?
That's pretty much how most brewers would approach FAN. Certainly, the different mashing conditions. So when it is used to produce the data that goes on a malt COA, can certainly change some of those numbers. We still measure the same parameters, but the numbers could change and could go up or down depending on the mashing conditions. Now, and similarly with starch hydrolysis, the enzymes that hydrolyze starch, all four of them have different temperature optima. So if you're mashing in at lower conditions or slightly cooler conditions, these enzymes have more time to do their job. Same with the proteases, the enzyme that hydrolyze the storage proteins. At lower mashing temperatures, these enzymes have more time to do their job. If we mash in at high temperatures, and this is the difference in sort of when we look at a malt COA, whether it's been used, the data has been produced from a Congress mash, which is a low temperature mash in, and then we ramp up to around 160 Fahrenheit, these enzymes still have an opportunity to keep working. So that sort of Congress or ramp mashing method is more of I would describe as a continued modification of the malt. This hot one-step mash in at around 150, there's only one or two enzymes that will be alive for more than about 20 minutes at that temperature. They are just denatured. One of those is alpha amylase. It keeps doing its job in hydrolyzing starch. The other one is actually a carboxypeptidase, and that's a special type of protein enzyme that will actually cleave off these amino acids. So FAN is one of those things we'll actually see increase in an infusion mash, a hot mash, because it has that optimal temperature. So we can see FAN levels higher in the Congress. We can see FAN levels higher in the infusion mash than we might see in a Congress mash, but all the other numbers might appear lower because all the other enzymes are denatured so quickly. So this is one of the differences between understanding how your malt COA was developed or the numbers put together for the malt COA. But that also has a commercial application and how it's relatable to breweries that might still mash in at relatively low temperatures, still giving those malt enzymes time to work. The kiln temperature in the malt house will start to slowly denature some of those enzymes, especially if the kiln temperatures are pushing up above 75 C, getting closer to 80 C. Again, those enzymes will start to denature. But we go into a cool mash, out of around 45 C. Sorry, I'm still sticking to my Celsius language.
All good. That's okay. Science podcast, we can stick in Celsius.
And those enzymes will still work. They might not be at optimal performance because they've been slightly denatured in the kiln, but they will still have some activity. So Congress mash or a low mash that might be stepped or ramped, you'll still get modification of these proteins. You'll still get a high level of FAN. If we just mash in at a hot temperature, then basically all the enzymes are challenged. Only a couple of them will survive for very long. And it does change the whole profile of what's in the wort. It changes the sugar profile. It will change your amino acid profile. And I guess that's probably one of the underlying issues, if we can say, we might have with FAN. It's a measure of total amino acids. It's a bit like when we measure gravity and report Plato. It doesn't tell us the profile of sugars. Yeah. And for some brewers, for a little bit more efficiency, maybe knowing your sugar profile would be better than just knowing your overall gravity. Same with FAN. We can report 150 milligrams per litre of FAN, but it tells us nothing about the amino acid profile. And yeast can be a little bit fussy, and it does like certain amino acids.
I didn't realize yeast could be, right? Certainly. I think most brewers would agree with that statement.
Yeah. And basically, the FAN method typically does not measure one of the most abundant amino acids coming from certainly the grain, which is proline. So the FAN method, which is used a ninhydrin chemical as the color forming compound when we do the method in the lab, it bonds to a particular molecule on an amino acid. In proline, the ninhydrin doesn't have access to that amine molecule. Huh. So when we report FAN, you could probably add 10 to 20% more to think, well, proline is part of that. Uh-huh. So that's one of the first things we think about. We can talk about, we've got 150 to 180 milligrams of FAN. That's enough. We'll add another 10 or 20%, and you've got proline. And then we have the whole conversation around what's the value of proline. Yeast doesn't assimilate proline, so it's left in final beer. Proline can contribute to certain beer hazes when it bonds to polyphenols and it drops out of solution in storage. So the downside to not measuring proline is not knowing the risk of some of the negatives that will come with proline in finished beer. But then once we sort of get past the malt process, and I think most brewers, if they read their malt COA, they're looking at three numbers. They're looking at extract, they're looking at colour, and they're probably looking at FAN. And when it comes to FAN, it's a case of, I've got enough.
Ding, I checked that box, right?
Yeah, I've got 150 to 200 milligrams per litre. That's enough. That works for them in the past. It'll continue to work for them, and that's fine. But we do know that there's subtle changes in the profile of amino acids coming from the different grain sources, depending on where they're grown, depending on the conditions that the grain is grown in. We do get variation in the profile of amino acids that form these proteins, that make these proteins. And climate change is a whole separate conversation, Cade, but I talk a lot about climate change at conferences.
Yeah.
But proline is one of those amino acids that's actually, we call an osmotic regulator. So it's actually expressed more when the plant is stressed because it helps deal with the stress. So as the world might get hotter and drier, we might see barleys and wheats that have higher levels of proline. Even though the grain still might have the same total protein content, there could be a shift in the amino acid balance. And as a consequence, in the brewing process, we could see some downstream problems. But when we measure FAN, we might still see the same number, 150 milligrams per litre, but we might actually have more proline present. So this is one of these things that I'm more than happy, and I think most people agree, let's stick with measuring FAN. We can't do much more than that at the moment because you started getting to amino acid analysis, and that's costly and time consuming and sometimes completely unrealistic in most applications. It's helpful for researchers to understand what's going on in a deeper level. But
yeah, maybe cost prohibitive for a brewer that's trying to look into
on a
it.
day-to-day basis, it's way out of the realm of any routine testing. And I think brewers would still like to be able to measure a fan on their finished beer.
Yeah.
Because that's possibly the next risk is what's going in the package.
Yeah, and I want to spend some time talking about that, like fan going into the package. But before we do that, I want to back up just a little bit because you made a couple of really, really great comments there. And so one of them was this whole concept of total fan, right? And again, you know, it's like the pie, right? The pie might be the same size, but the protein mix is changing depending on, you know, like you said, osmotic stress is a great example, you know, for climate change. And that's super important. But then again, the key thing here, a key thing here too is let's not throw the baby out with the bathwater, right? And I think that's also what you're saying is we still need to look at fan, right? Because fan can be an indication for the brewer of, you know, how much fan is available, right? Total fan. And it might be a different mix or proline or anything, and brewers might have had experiences in the past, but fan is still a useful metric for brewers. I think that's what I hear you saying. And my question, if that's right, I'll ask you if that's right. But then also my question is, so then what does fan do? You mentioned we need 150 or 200 milligrams per liter, but what does it do? Why do we need that? And where does that plug into the brewing process?
I would always say we need to be measuring fan. And it still will be really informative for brewers to think about how much or what level of total amino acids I have going into fermentation. It will change slightly because of the mashing and boiling process and the hops you add, and generally it'll go higher. So you'll always have more than enough amino acids. So we're basically jumping into fermentation and we're presenting the yeast with a source of nitrogen. So we present the yeast with a source of sugar to make alcohol. That's probably the primary reason. We want yeast to produce alcohol, change the sugar to alcohol. But to do that, it also needs a source of nitrogen. Like every living organism, it needs nitrogen. As two-legged animals, we eat protein as our source of nitrogen. But microbes can't eat large proteins. They eat really small parts of protein, so they consume amino acids. And depending on the profile of amino acids, then the yeast will assimilate these amino acids relatively quickly, usually within 48 to 72 hours. But then it becomes almost like a sustainable process of producing its own amino acids. During fermentation, it takes more than usually 3 days to complete fermentation. And during that total fermentation period, the yeast is going through this exponential growth phase in producing more yeast cells, consuming more sugar, and making alcohol. So during that growth of the yeast, it needs to be, you know, as the yeast is producing a daughter cell, it needs to be able to produce proteins that are involved in the organization and structure of the yeast cell, and then all these enzymes involved in these metabolic pathways. So there's this continuous consumption of amino acids to keep making proteins and enzymes. But after about 3 days, the yeast then has got enough amino acids from the wort, and it can start to look after itself. So we normally see that within 2 to 3 days, the yeast will have consumed most of the amino acids out of the wort. The fan numbers will drop really low. You never see a fan number at 0, but it'll drop really low. But then at the same time, as the yeast is producing some amino acids itself, it will excrete some amino acids. So often we actually see a fan level in finished beer higher than the fan level of the malt the brewers might have used.
Okay. Wow.
And this is where we're trying to still understand, what does that mean, that the fan levels are so high in finished beer? And I've had a lot of brewers from really big brewing companies say, what should our range of fan be in our certain beer styles? And I don't think there's a single answer to this.
Okay.
It will depend on lots of variables. But that's the overall purpose of us wanting to keep measuring fan, and the importance of measuring fan. But I think it'd be one of those things that we should be measuring it in the malt, but we should now be thinking about measuring it in the beer.
Yeah. Yeah, so I hear you saying that there's that fan is primarily a yeast nutrient, right? That it's providing yeast those building blocks that they need during fermentation. And actually, a quick aside, I heard John Palmer talking at the MBAA conference, and he had a good quote. He said, the yeast doesn't care about making alcohol, right? The yeast doesn't want. I mean, it'd be just fine not making alcohol, but it cares about making daughter cells. It cares about reproducing, and so it's doing those things to reproduce, right? It's consuming sugar to get energy. It also needs amino acid to build the structures of its daughter cells, and that's where those things are coming. So that's where yeast needs it. It's just consuming sugar and making alcohol to rapidly grow, right? And to rapidly expand. I thought that was a really good way of describing that. And that's kind of so what we're talking about here with fan. But ultimately, and maybe this is the wrong way to think about it, but it sounds like a fan is kind of a byproduct, right? It's sort of a byproduct of the malting process. It's a byproduct of mash process. It's a byproduct of yeast, of fermentation. And it kind of just sticks around through the whole process. It starts in the malting and the grains, and it kind of just hangs around. And so a question I have then is fan a good thing or a bad thing? And this may be a little bit time dependent. And it's pushing up against a break. So I'm going to go ahead and take a quick break, and then we'll start with that question. Is fan a good thing or a bad thing? So we'll do that, and we'll be right back.
Established in 1995, More Beer has been consistently serving the greater brewing community since the times IPA was expected to be bitter and clear, and there are reasons they've stuck around so long. In addition to their massive product selection and excellent customer service, More Beer has locations on both the west and the east coast of the United States, which translates into fast shipping times regardless of where you live. And when you spend more than $59, shipping is free. When you're in need of brewing ingredients and gear, there's no better option than morebeer.com, one of the most trusted shops on the planet.
All brewers share a common goal, which is to make great beer. And to make great beer consistently, you have to understand the raw ingredients, the process, your equipment, troubleshooting, and the ultimate use of the beer, whether it's to sell for profit, just to drink for friends, or just to drink with friends. Great beer is often the work of time and arduous effort. And today we're talking about fan. And so, Glenn, right before the break, I was sort of asking, what is fan a good thing or a bad thing? It seems, I made the comment, it kind of seems like like a byproduct that's kind of a byproduct of the malting process or getting at the starches. And then during the mash, it sort of increases during fermentation. The yeast need it, but then they also excrete it as a byproduct. So is fan a good thing or a bad thing?
Yes.
Yes, fantastic.
Would be one way to address that. I think it's a good thing, regardless of the level of fan we start with, whether we've got enough or too much going into fermentation. The yeast is going to do what it needs to do in terms of survival and reproduction, and as a consequence, it will make some more amino acids, which will be probably excreted into the beer. So we can end up with a relatively high level of fan in beer, sometimes more than the malt we started with. And that's all important because the amino acids are needed for yeast. But the negative would come when we could say it's a bad thing when amino acids can be responsible for some negative attributes in beer. So some negative flavor compounds are produced from amino acids. We know that valine is involved in diacetyl formation. Leucine is also involved in some higher alcohol formation. So this is where the amino acids can be both the good guy and a bad guy, depending on what compound. And this is where, as the yeast is growing and under certain conditions, whether it's pH of the wort, level of oxygen, other things going on in the fermentation, the yeast can actually decide to do different things and sort of upregulate different pathways. And as a consequence, it can use those amino acids in different ways. When it could be a really bad thing is when some amino acids are involved in staling. And this is when you might have a beer in a package and most brewers are hopeful that their beer will be consumed in a relatively quickly timeline within a month or so. But when you have beer in packages for months at a time, and depending on the storage conditions, then we can have staling compounds formed. And, certainly abundant amino acids like valine and leucine can be involved in some of those staling compounds. So this is where understanding the fan and knowing that a certain level of fan could put you at a reduced risk or a greater risk of staling. And then always sitting in the corner, tapping his finger or tapping her finger is proline. And going, I'm still here.
Yeah.
Don't forget about me. I'm still here. And proline can potentially then bond with some polyphenols and, again, cause some haze in beer styles where we don't want haze. But the downside is we don't measure proline when we measure fan. So we just have to assume that add another 10 or 20% will give you total proline and fan. So it's one of those things, and I've said it before, I think it's one of those attributes we should be measuring a little bit more of. It's a relatively easy assay in the lab. Most labs, certainly university labs, and if brewers don't have access to labs themselves, there's plenty of universities and colleges could probably do this basic ninhydrin assay. It's one of the ASBC methods. So it's a recognized method. It is a relatively straightforward method. I started doing these methods way back in the 1980s. It's one of the first things I learned, other than doing a Kjeldahl method.
Yep.
And it's just one of those things. So while we can talk about measuring all these things and we can be quite fanciful about let's measure more sugar, let's measure more stuff, the reality is it's not possible. It's costly and time consuming. But if brewers could get a baseline of fan in their beer styles and just track it from time to time, it might help them understand when they might have some sort of post-sales issues on quality. It might have come from some amino acids that we just weren't even considering the total amount of amino acids other than what's coming from the malt.
Yeah, and it's so interesting to think about how this breakdown is potentially, doing different things. Like I think you mentioned valine and diacetyl. That's the one that just pops to my mind really quickly. And leucine, I think, and higher alcohols. If these amino acids are sticking around, it seems to me is one of those things. It's like, I would really want to know what's in my beer, right? How much fan is actually in my beer, and that was one of my questions I was going to ask: is how is fan measured? And when we don't need to go into the nitty-gritty details, but it sounds like there's an assay, an ASBC method. And then the next question I was going to ask is where, if breweries don't have access to, fan method or ninhydrin or, any of those other components that are required for fan, where can they get this assay completed? And I hear you say, Cal Davis certainly and local universities would probably be welcome to, to send in your samples and have them run right there, or have them run at those universities. But then I have a question. So I've got my fan number back, and I don't know, let's say it's 200 milligrams per liter, to make up any number. What do I do with that information?
Just record that, and then probably do another batch in another few weeks, depending on how quickly you're turning your beer over and different beer styles. And then just record that and just have there for any possible troubleshooting that may come. For a lot of craft breweries, I think, especially if they're mostly taproom sales, they'll have turnover beer relatively quickly. I think some of these negatives that might come with some of the amino acids is to do with longer term storage. So if brewers are putting beer in packages and losing track of those packages because they're going across state or even across country borders, then they probably not necessarily have a lot of assurance on how that beer is stored. And if it's been looked after at all, is it in cold storage or is it going from warm to cold and back again? Then that's at greater risk. So it would be useful background information for brewers to have that and be able to relate that to if they're suddenly getting some negative off-flavor complaints about that. And then we could probably relate that back to possibly higher levels of fan. For me, high levels of fan in finished beer are a bigger problem than high levels of fan in malt. And I would personally think, depending on the beer style, things between 50 to 100 milligrams per litre in finished beer for long-term storage would be a safer option.
Yeah, but you mentioned that it might actually be higher than what's present in the malt because of all the process, a heavy dry hop charge, or a mashing regime like an infusion mash that's kicking you up a little bit higher, not giving you not letting time for the proteases to do their work, all of these things. So brewers could have, if 50 to 100 milligrams is sort of the goal in the package, brewers could have much more than that.
Easily. We've done some testing on some finished beer for other things that we've been testing for, and we thought, well, let's do fan as well. And I'm seeing, again, we say we, or I say I don't do any lab work anymore. I rely on my students and staff to do that. I have the luxury of having people to do that for me. But some of the beer that we've tested, we're seeing well over 200 milligrams per litre. Well, I would be shocked to think that brewery or those breweries are starting with malt at over 200 milligrams per litre. So depending on their malt load, if they're using other adjuncts, so solid adjuncts like rice or something else, the amount of hops they're using, if they're actually adding fruit for a sour beer, there's all these little extra doses of amino acids coming in that could be problematic downstream. And just one example, if I can, Cade, is to talk about sour beers. And I've got a postdoc here from Lithuania, Paulina, and she's been testing some sour beers for a particular off flavour. And we know that off flavour in sour beers comes directly from proline and leucine. So again, the level of amino acids is having an impact on the potential of this off flavour being formed in sour beers. That brewery probably doesn't know the level of fan it has to start with. So understanding your fan levels, even though it's not directly relatable to the profile of amino acids, generally if we think high fan, we have a lot of all the amino acids, and then there's greater risk than if you've got a low fan. So any fan finished beer above 200, I personally would consider risky.
Yeah. Yeah, it certainly sounds like. So then how do you let's start with that metric then. Let's say you're over, 50 or 100 milligrams. Are you just sort of SOL, or is there something you can do to reduce the amount of fan that's in your beer, or do you really need to go back and look at your process and figure out, maybe different mashing regimes or recipe or any of those things?
I think you'd have to go back to pretty much the start and look at your malt quality, look at your mashing conditions, and then looking at your hopping rates. And if you're doing a lot of dry hopping, as James's research showed, the amount of amino acids you're adding when you're dry hopping with yeast, if you're dry hopping after yeast, you're still adding amino acids, but the yeast won't have access to them. And then if you're adding other things, fruit or something like that in different beer styles, so there's also this constant dosing of amino acids. So I think for some breweries, they might like to just start testing some of their beers, but also testing during the process to see where there's an increase and decrease in these amino acids. And then understand that all of these inputs are bringing some amino acids with them. And eventually they might go, well, we might need to dial back on something if there's suddenly some sort of downstream negatives that are associated with amino acids.
Yeah, maybe dialing back, and then I guess is there are there opportunities for measuring fan, say, in raw ingredients like the fruit that you're adding or the hops themselves? Or obviously James has done work looking at amino acid contributions from dry hops.
Absolutely. The total amino acid assay fan is relatively straightforward. And you can measure it in most products that would contain proteins, because anything that's got protein had to have amino acids to start with to build those proteins.
Yeah, that makes sense. Yeah.
And if there's at any point, when the fruit or when the seed or when the grain is fully mature, there would still be some leftover amino acids, because the organism is making amino acids knowing they'll be used. But it might get to a point, it's still made enough amino acids that all the amino acids didn't get used. So we've got some leftover amino acids, some residual amino acids. And we see that in grain as well. We see leftover sugars in grain that weren't used in starch development or beta-glucan development. really tiny amounts, but they're present. So you could actually measure that in the grain. You can measure that in malt. You can certainly measure it in wort, post-mash, post-boil, post-fermentation. And that would give you a full picture of where your fan is increasing or decreasing during your process. And for most brewers, from hopefully all of the time, it's just data that they'll have at hand. But occasionally, if there's something to do with what's happened in the finished beer, then they might be able to go, oh, well, my fan level was X. And of course, because of that, then there might have been some amino acids that contributed to that negative flavor or negative quality product.
Interesting. Is fan level then like style dependent, or like is it higher in a hazy dry hopped IPA or versus a Pilsner, or?
It'd certainly be higher in most ales and IPAs. And again, depending on how the IPA is made and a hazy's made, if they're adding other raw materials to induce that haze, then that other raw material is probably bringing some amino acids to the party. So again, most brewers are only looking at the fan on their malt COA, and that's normally on their base malt. Most maltsters would not probably reporting fan on highly coloured malts or highly specialised malts. They might report extract and they might report colour. They might report diastase potential, even though those higher temperatures have basically killed off all the enzymes. You're more likely to miss getting fan data on highly coloured malts, but there would still be some amino acids left over. They haven't been used up. And then if we're adding other raw materials like rice as an adjunct or corn as an adjunct, they're bringing some amino acids, but also they're bringing proteins that the proteases are going to try and hydrolyze in the mash. So that's potentially a new source of amino acids given the right mash conditions. So it's one of those things, and it would be a long game for most brewers to start profiling their amino acid profile during the whole process. But I would hope for all of those brewers, it's just data to have in hand, and they never have to worry about any negatives that might come with packaged beer. But if that was the case, then the fan information might be able to tell them what's happened.
Yeah. So let's say then we've got a high fan. Where would you start in your process? So like, let's just say you've got a package that it came back at 200 or higher or something like that. What would be the first thing that you might change to see if you could reduce fan?
It would depend on the beer style, and certainly fermentation conditions, the yeast strain, because all of these things are yeast strain dependent and fermentation conditions dependent. Often you might want to go back to your raw materials and say, well, how much malt am I adding? My grist to liquor ratios, what else am I adding? How much hop am I adding in the kettle versus dry hopping? I guess one way to consider if you're dry hopping is then to start to use hop products rather than hop cones or hop pellets. That might reduce some of the fan that's coming in through that pathway. But certainly I'd be looking at all the raw materials that are going in, and most breweries wouldn't be looking at anything other than the malt in terms of what the fan profile might be.
Yeah. Yeah. So it sounds like expanding outside of just malt if you're trying to decrease fan in the brew house.
Yeah. If you're getting in with high fan levels, you need to be able to track that development of the fan through the brewing process. And again, it takes time, some cost involved. For some brewers, it might not be worth their trouble. Other brewers, it might end up being quite a saving in the long run, instead of having to do a package recall or having some negative complaints from customers. I understand that all the logistics around getting all those samples, getting them tested, all those sorts of things, and then maybe just sitting on data for years. But then it might be something in the long run that really does help you resolve a problem.
Yeah, I mean, something popped into my head here, and I have no idea if this is even related or not. But one of the things that we talk about all the time is the shelf stability of hoppy beers, of those hoppy, hazy IPAs being very poor shelf stability, right? They're just not. You want to drink them as fresh as possible, and you don't want to let them age for a month. I'm starting to wonder, is this an interplay with, is it because of the hops? Is kind of what I thought. Is just you're throwing in a whole bunch of hops, and that's causing shelf stability issues. But maybe it's this interplay with hops and fan that's causing some of these shelf stability issues with those. I don't know. Are those two things related, or is that potentially why some of these, heavily hopped IPAs don't last as long because the fan content is just getting all wacky?
That might play a role. But there would be other things that I think would be more positive in terms of providing some stability with the level of antioxidants, the level of alcohol, those sorts of things. But in amongst all of this is the amount of dissolved oxygen as well.
Of course, yeah. Mm-hmm.
So how much DO, and I know a lot of brewers are measuring DO, so they have a really good understanding of the level of DO in their finished packages, and they're all targeting, I'm guessing, less than 100 ppb. So understanding that, and then if your DO is right, then there's something else going on, and this is where they could start to look at these other compounds that can contribute to staling, other than is the package perfectly sealed. So is it coming from bottles or is it coming from cans? Where's the risk of staling or any oxidation? So again, they'd have to do some homework. But if they're measuring DO, they could probably eliminate that as a negative. And then there's something else coming into the equation.
Yeah, and which could be fan. That's interesting. And so another place that you had mentioned earlier about potentially reducing fan is changing your mashing regime, is also doing a protein rest. And one of the questions that had sort of popped into my head too, thinking about Sophie's research with glucanases and xylenases, right, the breakdown of those proteins, does use of those enzymes increase or decrease? Or let me just ask the open-ended, how does use of those enzymes like glucanases or xylenases during mashing, how does that impact fan levels?
If you've got good modification, so the glucanases and xylenases have worked really well, then you've potentially had more of your storage protein exposed to the proteases. So the level of modification, which is basically a measure of how much of the storage protein has been hydrolyzed, a good modification, we'd always get higher fan levels. So the risk is you could wind back your level of modification in the malt, but then that would bring some other negatives, because then you would have potentially more beta-glucan or aminoxylan. So this is where you could use a less modified malt, but you could then add beta-glucanase into your mash. And this is where using less modified malt, but then adding specific enzymes into the mash to target one group of one substrate, you really want to limit the hydrolysis of your storage proteins.
Mm-hmm. Yeah, that's interesting. So, okay, so I see that. So sort of using these as taking an undermodified an undermodified malt, which doesn't have those storage proteins broken down yet, and then adding the glucanases and xylenases so that you don't have your lautering issues and stuck sparges and all that sort of stuff, and still access to your starch, right? Because again, you have to break through all of these things, the glucanases and the xylenases and the storage proteins, to get to the sugar. Okay, well, that's interesting. And then I guess this might be, I mean, I don't know if this is a silly question or not, but would you ever have too little fan? I mean, is that something that's like a realistic problem for brewers, that they would have too little?
I think it is a possibility. I would be surprised that maltsters would be supplying malt with a low level of fan, right, unless the brewer has specifically asked for something with low. But any modification, greater than, in terms of Kolbach index of 40, even 39, you would normally have at least 100 to 120 milligrams per litre. It would have to be really poorly modified malt to be less than that. And the maltsters don't want to make that sort of malt anyway.
Right. Yeah.
So if you're just buying low modification, it's probably around 39, 40 on a Kolbach, you're still getting around 120 milligrams per litre. When you're above 40, you're probably getting close to 140, 150 milligrams per litre and well-modified malts of 45, 46, 47. Cold bark, you're looking at 160, 170, 180, possibly up to 200. So I think it'd be real risk in buying any malt that's got a fan less than 100, because you've got a lot of other stuff that hasn't been hydrolyzed, and it could become really problematic in the mash.
Yeah, really problematic in the brewing process. So that sounds right. What about adjuncts like corn or rice? Do those come with fan additions, or are those sources where you could dilute fan out if you needed to?
You could possibly dilute fan out, but then on the mashing condition as well, because the proteases that will hydrolyze or break down those proteins are relatively sensitive to high temperatures. So most of the proteases are denatured above about 55 Celsius, certainly 60 Celsius. If you present those proteases with adjuncts, most of the grain proteases are somewhat non-targeted in terms of they just see proteins coming from the grain. So they will still try and hydrolyze these storage proteins from rice or corn. So given the conditions, you could actually reduce the amount of fan with those sort of adjuncts mashing in at a higher temperature, because the proteases from the malt won't get a chance to hydrolyze those.
Interesting. All right. And then another question then about fermentation nutrients, right? So I hear all the time people adding yeast nutrient, which includes a big portion of fan, at the time of fermentation. And I guess there's kind of two different reasons or rationales here, right? I mean, you've got, fresh yeast that's straight from the manufacturer that's probably going to be packaged with sufficient fan and nutrients, that it needs in order to kick off during the fermentation, right? If we're using fresh yeast.
Yeah, we do need to add the nutrients, because the nutrients are bringing more than just free amino nitrogens. There's actually minerals and some other components that the yeast needs, that generally most worts will have everything it needs, but it's really just an assurance to give the yeast every chance it has, or every chance possible, to do the best job it can do. So I do know some brewers that don't use yeast nutrients at all, because they know there's enough coming from the wort itself. So yeast nutrients are always good, and depending on your yeast supplier, they might come with the package itself. But other breweries, and I know that we'll actually add separate yeast nutrient when we've got our brewing classes. We still use dried yeast, so we know there's some components coming with the dried yeast. So it's really horses for courses, and what the brewers want to do. They'll understand their yeast, they'll understand their beer styles, and they can add it accordingly.
Yeah. All right. That sort of makes that makes a lot of sense, right? That it's kind of, choose your own adventure in that way, especially with yeast and fermentation. But yeast still need the other nutrients. That's one of the key things that I hear you say there, is that there's other things than just fan, that whenever you're adding yeast nutrients. All right. So one other question before we sort of wrap things up: if brewers wanted to know more about fan, like if they wanted more resources or anything, or if they wanted to do, fan testing, where can they go to find that information?
I guess there's certainly a lot of publications that, I don't want to promote the book Charlie and I just published, but that might be a useful source. Right. But there's certainly a lot of great publications through ASBC and Master Brewers Association that can sort of give them a really in detail story of fan, what it is, the purpose and the value of different fan levels. But other labs like Hartwick with Dr. Bettenhausen's lab, Harmani, is doing a lot of routine malt testing, but the fan assay is applicable across wort and beer, and other labs that routinely testing malt and beer, they could easily do fan. So it's just a case of probably brewers thinking about who's their local college or local university. Is there anyone working in food science? Certainly if they're working in brewing science, they'll have a good understanding and access to the ASBC methods. That would be a great first port of call.
Yeah. Yeah, and it sounds like maybe this is something that brewers might be more interested in looking into, especially in your final beer, not just relying on that malt number. All right, well, we spent a lot of time talking about fan, and wow, we covered a lot of ground. I mean, I can't imagine. There's probably going to be lots of questions from this episode just over the things that we discussed. But if you wanted brewers to take away just one thing from today's episode, what would it be?
I think the thing for brewers to think about is there is fan in your finished beer. And knowing the level of fan could actually be an assurance to say you've got a stable quality product. Without knowing that, then you're probably at a slight disadvantage to know the not knowing any possible downstream risks in your packaged beer. So again, it's just one of those extra things we could be testing, and it's probably one of those things I would suggest over other things to test for. But understanding the level of fan in your beer and how that number was derived, where was all those amino acids coming from.
Mm-hmm. Yeah, and ultimately, sometime in the future, maybe we can actually make this, protein breakdown analysis, a little bit more realistic, or maybe cost effective, and go down that path too, so we can start to understand, how different proteins impact, especially the storage and shelf stability.
Yeah, it's probably opportunity for another chat in the future, Cade, but we've been actually measuring the total storage protein in comparison to the total protein. And the range we see is between 40 and 70% of total protein is the storage protein, and how that relates to the amount of amino acids presented or provided in the mash is significant. So again, we've used total protein as a measure of quality, but again, it's only telling us a really tiny part of the story. Measuring total storage protein, actually, I believe, would be more informative.
Huh. Interesting. Yeah, the actual storage proteins, the guts. What did Sophie describe it? She described it as like the tissue paper, the wrapping paper that's in there with the starch. Yeah. Yeah. Well, good. Well, on that note, and on the promise of a future episode to talk about this, I got you on the hook now, Glenn. Is there anything else you wanted to talk about fan that we didn't get to today?
I think we've probably covered enough for one episode. It might be something worth circling back, especially if I get some feedback from brewers and I get some more data that we can tie in some of the fan data with some of the overall quality, and if any brewers get any sort of negative quality coming out of beers that have high fan, this might be an opportunity to do some in-depth studies with some brewers if they're keen to see what this story could lead to.
Yeah, well, brewers, if you're interested, reach out to Glenn or reach out to me, and I'd be happy to share Glenn's information with you, or at least, assuming he's okay with it, I'd be happy to connect you. We'll say it like that. Yes. Yeah. And Glenn, thank you again, of course, for joining me in the Brew Lab.
No worries. Always great.
All right. Well, next week, Jordan and I will be back applying the science of this episode, and we'll see y'all then. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.